Welcome back. This is topic 7, so lecture 7, and the second part of metabolism and energy change in part 1 of your BIOS form. I want you to be a little bit comfortable with Gibbs free energy. We'll talk about it in review, especially if it's new. I would like you to be comfortable with exergonic, catabolic, and anabolic. And I think the best way to do that is just to remember which one goes with cell respiration, which one goes with photosynthesis. That was our practice question. We'll cover it again in review. The answer is 1 and 3. I think the way I would approach this if I were you is to try to make a table of Gibbs free energy and put anabolic, catabolic, endergonic, exergonic in the table and see how those words go together. Maybe give them some good examples besides cell respiration and photosynthesis. I keep forgetting which one I give you. We're going on with this, and I want to focus this part on ATP. I've had a couple questions because some people are comfortable with ATP, some people have just heard of ATP, and some people have never heard of ATP. So I want to give a whole intro and description of this molecule. It's incredibly important to us. Cells use ATP as the molecular form of energy that cells use to carry from one place to another. So if I want to make something happen, I want to put energy into a reaction, an endergonic reaction, one that requires energy, I need to use ATP. Your cells can't use fire. They can't use just straight up sugar. Sugar is too complicated a molecule. So you have this tiny little molecule that you use to drive your chemical metabolic reactions. This is a picture from your textbook of an ATP. I'm never going to ask you to draw it, but I want to show it to you. It has a ribose here. Os tells you that it's a sugar. And attached to the ribose is the nitrogenous base adenine. This right here is essentially a nucleotide. I'll show you a little bit more when we get to the DNA lectures. But for now, you can just, in my notes, I just put a big A there and don't worry about the molecule. Because what I care about is these. There are 3 phosphate groups attached to my adenine and my ribose. What do you know about phosphate groups? What's their polarity? Are they polar or not polar? Are they hydrophilic or hydrophobic? What about, are they acid, base, or neutral? And are they charged? This phosphate group is a charged, acidic, polar, hydrophilic functional group. And here I have 1, that's the alpha. This is A, B, and C. My 1 group, my 2nd group, and my 3rd group. They all have negative charges, and if you remember, opposites repel each other. If I try to put 2 negative charges, they're going to push each other away. So what happens is this bond here between the last phosphate and the second to last phosphate is what we call spring-loaded. It's really easy to break, but it has a little bit of energy in there. I got a better drawing. I don't know if it's a better drawing, a simpler drawing. Here is my ribose, my adenine, and these are my 3 phosphates. You might want to put some minuses there to remind yourself they're charged. My ATP, it stands for adenosine. That's this. Triphosphate. 1 phosphate, 2 phosphate, 3 phosphates. Adenosine triphosphate. When I want that bond to break, I do what we've done all along. I dehydration synthesize it. No, I'm lying. I hydrolyze it. Don't dehydration synthesize it. I hydrolyze it. And I cause that bond to spring-loaded bond to break. It will break easier than this bond because this one is negative and this one is negative and they're pushing each other away. The little bit of energy that we were counting as Gibbs free energy that was in this bond is released. Now the trick is to release it in a way that you can use it. You don't want a little explosion. You want this energy to go towards doing work. So kind of as a side note, thought note, how do you think I build this molecule, this adenosine, this part, with its 2 phosphates? It's an adenosine diphosphate, 1 phosphate, 2 phosphates. How do I build this molecule back into an ATP, an adenosine triphosphate? You know this. We have only had 2 reactions, and I kind of ruined it in the earlier part of my lecture because I told you we're going to dehydration synthesize it together. Now, this is going to involve a lot of energy and enzymes. I'm going to have to build this in. I'm going to have to take energy and this molecule and this one. We call it a phosphate group with a little I because it's going to be an inorganic phosphate. I'm going to put an enzyme, a little bit of water, maybe a lot of enzymes, and I am going to build back my ADP plus a phosphate with some energy, add water, and it's going to rebuild my ATP. It is dehydration synthesis, but this gets a special name because I am doing something that is so important that we call it its own idea. I'm putting a phosphate group on to this molecule. And when we build a phosphate, we call it phosphorylation. This process of adding a phosphate wherever I put it, every time I smack a phosphate onto something, I'm phosphorylating. This phosphorylation and the hydrolysis of ATP to ADP and back to ATP is a cyclical change. And right now, I'm going to show you how we're going to go back. This is the simplest version. We're going to get much more complicated than this. But here I have an exergonic reaction. Energy is released as I break my phosphate off. I hydrolyze it and I release a little energy. And then I can use some enzymes, add energy in to maybe the energy you took from food. I take my ADP, a phosphate, and some energy, and I phosphorylate the ADP back into ATP. This side of the reaction is endergonic. And we see this relationship all over the place. It's not just going back and forth between 2 of the same kind of molecules. Every time I break down ATP, the reaction is going to be exergonic. I'm going to release a little energy. That energy has to go somewhere. That's the point of creating this molecule for you. You don't want to explode. You want to push that energy into a reaction and make it happen for you. You want to run your metabolism. So we're always going to find the breakdown of ATP coupled, paired, kind of like holding hands, with a reaction that requires energy, a reaction that is endergonic. So, and I'll put this link in the bottom. This is showing how energy works with some proteins. I always imagine from high school, I know you do too, that as ATP, we break it down to do energy, this is what happens. Right? So the Kamehameha, like Goku's attack, right? He builds up energy. Okay, he's like spring loaded. He's ready to let go of all that energy, and then he just shoots it in the general direction. It's great, but it's all going into chaos and light and heat, which is what he wants. But this is not driving work. It's awesome, but it's not driving work. You can't use your APP like Goku Kamehamehas. You need to use your energy without blowing up in a controlled reaction to deal with your metabolism. You do something more like this. I'm sorry, this is less exciting than Dragon Ball. You're going to phosphorylate something with your ATP. ATP breaks it when it's broken, is an exergonic reaction. It breaks down and some energy is released. And what happens is as that ATP is broken, energy released, and it drives that last phosphate, the third phosphate that was on that spring loaded, into a protein or into a molecule. And just for a second, this molecule or this protein becomes an intermediate stage that we don't normally show when we write out the reaction, but it becomes a phosphorylated intermediate. And that phosphorylated intermediate is shaped differently. It now has a phosphate stuck into it. That new phosphorylated intermediate, that new shape, means that it is a little bit easier and a little bit lower energy to do the reaction that we want. It often means we just gave it enough energy to cause the reaction to happen. In the case of a transport protein, that may be causing it to open and close. Phosphate on, phosphate off, phosphate on, phosphate off. And each time a phosphate goes on, it uses an ATP. In the case of, this is one of my favorite proteins of all time. I drew it out for you. This is called a motor protein, and it moves like this. It's holding a vesicle. This is how we're going to move that container that's moving proteins through your cell. This little moving protein is like this with a vesicle on top. Phosphate on, the phosphate pops off. Phosphate pops on, phosphate pops off, and it walks. It changes shape. The protein or the reaction that we want to add energy to is coupled. We say that they're coupling to the ATP. ATP is exergonic and provides the energy. The reaction that we want to do is endergonic, and it requires that energy. So I drew this myself because I love these things. So this is a motor protein, and it's carrying a vesicle. We want to have it do work, but we can't just do this. We don't just shoot it like Goku and it walks. We need to create a phosphorylated intermediate. So what I do is I take my protein, my purple motor protein, my body gives it a little bit 1 ATP. The 1 spring-loaded phosphate phosphorylates my protein. ATP is hydrolyzed, and it gives its phosphate group to protein. The protein now here is a phosphorylated intermediate, and that causes it to change shape. The ADP is done. The phosphate falls off, and we go back to the original shape, where it then can take another step. Phosphate on, phosphate falls off. Phosphate on, phosphate falls off. And it walks down your cell, carrying that protein in the vesicle so that you can go through exocytosis at the end. This is what it is. I pulled this from a paper. I was going to just give you the paper, but I think it's a little too complicated. These things walk like little legs, and we talk about it going through a change in shape in response to being phosphorylated. We talk about it by going through a change in shape by being phosphorylated. ATP is hydrolyzed into ADP and the phosphate. That phosphate group binds to my molecule. It creates a phosphorylated intermediate. This is a shape change. And we call it a conformational shape change. It changes shape, and that makes it do something. Changing a protein shape, changing a protein shape causes it to change its function. And so we make it do something functionally by giving it a little bit of energy and causing it to go through a shape change. With that, I'm going to leave you to think about that. I want you to think about how you used to consider the function of ATP. I want you to draw me a very simple A with a bunch of phosphates on it. We are going to use cell respiration to build ATPs. We are going to use ATPs in photosynthesis. I want you to get comfortable building and breaking them up so that we can use them in our later reactions. And I'll see you in the next video. Have a good night. And the next two are very short, but I think you'll enjoy them. All right. Bye-bye.